everybody, and welcome to the muscular system, which is chapter 6. We'll be looking at the functional and characteristics of muscles, how they work together, the energy required, and different types of muscle cells that actually exist in our body. So some characteristics that they all share: they are excitable, they can respond to a stimulus. This would be like a nerve impulse. They are contractile, so when you actually take your biceps and you bring your fist to your shoulder, your biceps brachii, that muscle right there on the anterior part of your arm, is actually shortening. They can stretch. You can take that fist and move it away from your shoulder, and you can stretch that biceps brachii. And they can return to their original length after being shortened or stretched, kind of like a rubber band returning to its original size. So these are characteristics of all muscles. There is 3 types of muscles. We have skeletal, cardiac, and smooth. Throughout this lecture, we're spending our time on skeletal muscle. In future chapters, you will cover the other types of muscles. All right, so you have over 600 types of skeletal muscles. Some work together to cause a certain type of movement. So when you move your arm in a circle, it's a group of muscle that will participate in that action. There's some muscles that work in opposition to each other, so they're antagonistic. So your biceps brachii, it flexes your elbow. Flexion of your elbow is bringing your fist, your palm of your hand, to your shoulder. Now you can take that fist away from your shoulder, and that would be extension. So your biceps brachii would do flexion, and the muscle that would do the opposite of that, extension, would be your triceps brachii. So that would be an antagonistic muscle. And you can see there's an example of that. Here's a group of muscles that you guys will be covering in your lab course. So this is a good reference image when it's asking you about the actions. Okay, so muscles are going to attach to bones. They're going to move bones when they contract, specifically skeletal muscles. And they attach to bones for the most part via a tendon, which is just basically sort of this flat or even round. It can be various sizes, but this band of connective tissue that connects that specific muscle to that bone. There's a start point of the muscle, which is called the origin, and then there's a stop point of the muscle, which is called the insertion. The insertion point is the part that actually moves, so that's what causes the muscle to shorten. You can always remember when you're thinking of movements, insertion moves towards origin, and we can discuss that action in lab. Inflammation of those tendons is called tendinitis. Anytime you see itis at the end of something, it usually means inflammation of whatever the prefix is. And so sometimes tendinitis can lead to full tears. A lot of times rest is good. Tendons, just because of the type of tissue that they're made of, this dense, regular connective tissue, they have poor vascular supply. So if you don't get a good quality healing time, they could heal very poorly. And so you could, in the future, have to replace some of those tendons. Okay, so a muscle strain. So we brought this up in the previous lecture. There's a sprain and then there's a strain. A sprain is a torn or injured ligament, and a strain is a muscle or tendon type of tear. All right, so muscles, I'm going to go right to this picture here. So muscles are made up, they have these connective tissue sheaths that surround them. And so you can see here's the tendon. We have an outermost connective tissue sheath called the epimysium, okay? And then we have these fascicles. So all these circles right here, these are all called fascicles. You can see one large fascicle has been pulled out for your view. And perimysium, this surrounds each of the fascicles. Then we have what's called a muscle fiber. Now, this is a muscle cell. There's a plasma membrane. It has organelles like mitochondria, all right? But a muscle cell is called a muscle fiber because it's a really, really long cell, all right? And this also has a connective tissue sheath called an endomysium. Okay, so epimysium, that surrounds the entire muscle that you would see with your naked eye. You can maybe see fascicles. All right, fascicles, these are surrounded by perimysium, all right? And then the muscle fiber or muscle cell, that is surrounded by endomysium. So it goes epi to peri to endo, from outermost to innermost. Okay, so like I said, a muscle cell is called a muscle fiber. When we look at them under a microscope, and go right back to this picture here, you're going to have these tubes that exist inside a muscle fiber. They're called myofibrils. And guess what? Within them, you're going to have myofilaments, okay? They're called myosin and actin filaments. So there's thick and thin. Always remember, actin is thin. All right, so let's take a closer look at this here. And what we can see here is we have this thing called a sarcomere. We have this thing called a sarcomere. And you will have a sarcomere all the way. It's kind of like boxcar. So you'll have a sarcomere over here. You'll have another sarcomere over here. You'll have another one over there, and so forth. And so all along the muscle fiber, you have all of these sarcomeres. And the borders of a sarcomere are Z disc to Z disc. And within that sarcomere, we're going to have our actin filaments and we're going to have our myosin filaments. Our actin filaments, they are the thinner ones right here. And then our myosin filaments, they're going to be the thicker ones there. Now, the structure of this actin, if you look over here, the actin filament, okay, it has some protective strands over them. This is called troponin and tropomyosin. Sometimes they call this troponin-tropomyosin complex. And what this does is it normally blocks actin, okay? Now, the myosin has these myosin heads. Kind of looks like a two-headed golf club there. Now what's going to happen is these myosin heads, which you guys can see here, these myosin heads will grab onto the actin and it's going to pull this actin molecule this way. These heads over here, they will pull on these two actin strands and they will drag it this way. Now this actin is attached to Z discs on each side. If the myosin heads grab onto the actin and pull them towards the center line, the whole sarcomere will shorten. And this is how your muscle contracts. Now, our muscles can't be contracting all the time. And so that's why we have this troponin and tropomyosin complex that blocks the actin from the myosin heads from binding. All right, so we will remove this complex. Calcium will actually do that. Calcium will remove this troponin tropomyosin complex so the myosin heads can attach on. And the myosin heads, they will require ATP. They will actually require ATP. So both calcium and ATP is required for a muscle contraction. Let's take a look at some other images. Okay, so looking right here, you have your muscle cell. And within that muscle cell is going to be your filaments. And you can see your filaments here. You've got your actin. And that actin is directly attached to the Z disc or Z line. And then you have these myosin filaments. And they always change the colors in them. The actin is also known as thin filaments, and the myosin is always known as thick filaments. And this is what gives that characteristic striated-like appearance when you look at muscle, skeletal muscle, under a microscope. Let's look at some more pictures here. Oh, there is some great videos on the Pearson site for videos of showing how this works here. Okay, so looking right here, this is showing you the myosin. This is in a relaxed muscle. So here is one sarcomere. You would have another sarcomere over here, another sarcomere over here, another one, and another one, and so forth. You can see that we are relaxed, but when the myosin heads do grab on, you can see in this view below, that pulls that Z disc from each side towards the center line here, and the whole sarcomere is shortened. And that is how you contract a muscle. This is called the sliding filament theory. So this is a video here that goes over that. This is showing you how we have your troponin tropomyosin complex, and you can see how it is blocking. You can see how it's blocking these sites on the actin, and those sites are normally for that myosin head to grab onto them. But why are they being blocked? Well, we can't have a muscle contraction all the time, right? And so you can see in the presence of a calcium ion, this troponin tropomyosin complex slides off and the myosin head can bind on. Now that myosin head, we'll talk about in a minute, that will require ATP to grab up. And, oh, I don't have a picture of that. I thought I did. Okay, so there is ATP binding sites on this myosin head, and ATP will be required for this myosin head binding. Now, what specifically actually happens is that in a previous cycle, we prepared the ATP and we broke it down into adenosine phosphate plus an inorganic phosphate. And so what gives it the energy to put the head up there, when the head of the myosin, when the myosin head attaches onto the actin, it's called a cross bridge. And it releases that phosphate and releases that ADP. Now a new ATP, will attach onto the myosin head, and that will allow that myosin head to detach. If you don't have ATP, like when you're dead, okay, that myosin head cannot detach. It stays attached to the actin, and you get what's called rigor mortis. Eventually, that will subside when there is tissue and cell death. Okay, so calcium ions, they come from an area called the sarcoplasmic reticulum. It is basically the smooth endoplasmic reticulum in a muscle cell. And transverse tubules, which are connected to the plasma membrane, carry electrical impulses to this sarcoplasmic reticulum, telling it to release. So you're going to have an electrical impulse, and we'll show that in just a second, that will travel down the T tubules, and that will cause the sarcoplasmic reticulum to release calcium. And remember, calcium is required to remove that troponin-tropomyosin complex. Okay, so where the nerve meets the muscle, this is called the neuromuscular junction. So this is an axon right here, which is found within a nerve. There's many axons within one nerve. And in that axon, it will release neurotransmitters called acetylcholine. So there's these little vesicles in there with these little chemical messengers. That's these little purple dots. And that chemical messenger here with skeletal muscle is known as acetylcholine. Acetylcholine travels across this area here and binds to the muscle cell membrane. And when it does that, it causes an action potential to travel down the cell membrane. Now that travels down the T tubules, and then that causes the flooding out of calcium. So you can see the flooding out in calcium. So this electrical impulse is what causes the release ultimately of calcium from that sarcoplasmic reticulum. And once that calcium is binded to the troponin-tropomyosin complex on the actin, the myosin heads can attach on. Remember, the calcium allows that troponin-tropomyosin complex to remove from the actin, so it allows the myosin heads to attach. Remember, the myosin heads do require ATP in order to attach. Okay, so just a disease out there. It is a sex-linked disease, so it's more commonly found in males. There's many different types of this inherited condition called muscular dystrophy, one of the most common being Duchenne's. And basically, there's a protein within the muscle cell. We didn't really see it on the images. It's called dystrophin. And basically, dystrophin is faulty here. So they lack the gene for the production of the normal protein dystrophin, and basically the muscle cells, they deteriorate. So okay, so I mentioned that neuron. Okay, I mentioned that neuron there contains right here. It's within a nerve right there. So you can see the nerve coming from the spinal. This is a motor neuron, okay? And so if you look at this definition, we have this term called a motor unit. A motor unit is a motor neuron and all the muscle cells it's stimulating. So here is a motor neuron right here, and you can see it's stimulating multiple muscle cells. Okay, so this is a motor unit. And when you have a motor unit, okay, you can attract more motor units. So not only can one motor unit contract, you can actually recruit more and more motor units. And the more motor units you recruit, so there could be one over here, there could be one over here, there could be one over here. The more motor units you recruit, the stronger the contraction, okay? And so that is called recruitment. Now, there's a term called a muscle twitch. And this is a single stimulus. This is not something that you would see like when you think about someone doing a twitch. This is something that would be read in a lab in what's called an EMG, an electromyogram. And so when they stick a needle in a muscle and they look for the responses, okay, they're reading a muscle twitch, right? It's reading a muscle stimulus, all right? And basically, if you get another muscle stimulus, if another stimulus is received before it has time to relax, the second twitch will be stronger. And basically what happens is that there's less time for the calcium to go back into the sarcoplasmic reticulum. So the calcium's already there. The troponin tropomyosin complex is already exposed, the actin. And so it just makes the contraction happen faster, more powerful, and quicker. And you can see here, we're looking on this side right here is the strength of the contraction. And this is looking over time right here. And this is looking at a single twitch. And this is looking at multiple twitches. And look what you're seeing. You're seeing it has less time to relax. And so you get a stronger and stronger contraction. This is kind of like the basis of a pre-workout, so to say. And you can see here we can get a full sustained contraction too as well. This is called tetanus until fatigue would set in. All right, so when we talked about fatigue, that means a muscle needs energy. So we get energy in our body in the form of ATP. We do store ATP in our cells. We also store another molecule called creatine phosphate, which can donate this phosphate. So when you use ATP, you cleave off a phosphate, and now you have ATP plus an inorganic phosphate. You use that phosphate, and now it's done. It can't be used. So we need to go through what's called cellular respiration to replenish this ADP right here. But we can donate a phosphate from creatine phosphate too as well. Now, also what's unique about skeletal muscles, it can undergo what's called anaerobic, which means without oxygen. You don't make very many, this is basically going through glycolysis only. You only make about 2 ATP here. Aerobic respiration, this is going to be something where you're going to have plentiful of ATP. You're going to be using oxygen. So this just references some terminology that we discussed previously in our earlier lectures. And you can see here that during over time when someone's running, you can see we've got ATP that's stored here in the first beginning, quick 6 seconds. You can see ATP from creatine phosphate and ADP. You can see ATP that's generated from glycogen. We actually store, glycogen is the storage form of glucose. So we do store glucose in our muscle cells, all right? And so you can see here that we have ATP is generated from that glucose that we have stored in there. And then there is some oxygen replenishment after the exercise there. Now there's 2 types of muscles that exist in our body. We have slow twitch and we have fast twitch muscles. Slow twitch is going to be muscles that they're less likely to fatigue. They contain a lot of endurance, a lot of mitochondria. Think about when you get a rotisserie chicken. This is like the dark meat. It has a rich blood supply. It's very dark in color. The dark meat is found on the wings. Now, I know chickens don't really fly, but if it was a bird, this would be found in the wings. And the wings are very resistant to fatigue because otherwise birds would be falling out of the sky there. So this is your endurance type muscles. This is like your jogging muscles. Someone who is a marathon runner would have a lot of slow twitch muscles. I can't say that word today. Fast twitch muscles, these are going to be more powerful. They're going to be stronger. This is going to be like weightlifting muscles, but they fatigue very quickly. So they use anaerobic pathways and fatigue very quickly. So a bodybuilder would tend to have more fast twitch muscle cells. And over time, you can change your muscle cells from slow twitch to fast twitch. All right, so aerobic exercise is going to be things that provide enough oxygen so you can maintain ATP for long periods of time. Lots of mitochondria, usually don't get very bulky, so bodybuilder-like, whereas resistance exercises, this is going to be more weightlifting, and so the muscle size is increased here. Okay, so that is it for Chapter 6.